Hola, muchas gracias por unirse a la segunda sesión.
Hi, thank you very much for joining us to this second session of our symposium. We are going to continue in a different format, but we are also going to continue with our talks. And in the end, we're going to conclude with some discussions, some conversations. So now I'll pass it over to Ixchel, who's going to be our next moderator. Please remember to keep your microphones muted so we don't have any interference, and just turn them on when you have any questions, whether in English or Spanish. Thank you very much. Over to you, Ixchel. Hi, good afternoon. Thank you for joining us this afternoon. My name is Ixchel Gonzalez Martinez, and I am part of the Eco-Evo group in Latin America. Today, I'm very pleased to introduce Valeria Carreira. Valeria is a professor at the University of Buenos Aires, and she is also a researcher for one of the technical and scientific groups in Buenos Aires, where she works with the species of Drosophila and her relationship with her flight capacity. Today, she's going to talk about the flight capacity and its relationship with the morphology of a specific species. Please be welcome, and all of you, to our dear audience, remember that you have the Q&A session to ask your questions. Thank you very much. I'm going to share my screen with you. Can you see my screen? Can you now see my screen or not yet? Not yet. Now we can see it. Yes. Thank you very much. Yes, it's a bit strange having two screens for a single presentation, but thank you. Thank you very much. All right, so good afternoon, you all. Just as our colleague has just said, my name is Valeria Carreira, and I was honored to receive this invitation to conclude such an important symposium for the study of evolution. To be honest, I'm completely honored to be joining you today, and I hope I can meet your expectations. So to be honest, this is the very first time that I run a conference, that I lecture on something. So I'm going to tell you some of the issues that I had in my career as to promote, let's say, this sort of researches. So that's why Henry was saying that this is, let's say, that here we have a different modality for this second session. And maybe that's why I think this is not going to be a quite academic, let's say a quite conventional academic lecture, but I just want to hold a conversation with you. So just to get started, I'd like to get started by telling you that I'm the oldest kid of a family, a very, let's say, a very big family of, can you see my second screen? My second slide, I'm sorry. Yes, we can. Okay. So as I was saying, I'm the oldest kid in a middle class family. So that's why I attended different schools, and it was very difficult for me to conclude my studies. So I ended up studying biology, given that I thought that there was a broad variety of topics that I could study here. And I started to study evolutionary studies because I think that evolution can cover pretty much every single aspect of human life and life in general terms. All right, so as I was introduced, I've got a bachelor's degree in biology from the Buenos Aires University, and I'm also a professor since 2004 in the same university. I'd like to highlight that this teaching experience. Likewise, I'm also a researcher for the Technical and Scientific Center of Studies in Argentina from the same university. And actually, I became part of this group the same year that I became a mother. So as you have also said before, as it was said before as well, I've been studying different adaptive characteristics for the last 20 years. And I have especially worked with morphological characteristics in flies from different species, raised under different environmental conditions. And in recent times, I started to study the relationship of these characteristics and their flight capacity. this given to the different bachelor's, let's say, research projects that I was part of, and the work that I conducted together with some other colleagues and this also, of course, ended up with different publications and different academic products in different journals. So with that, I'd like to introduce you to the most academic part of this talk, which has to do with this flight capacity and its relationship with morphology in Drosophila, which is one of the latest topics that I've been working on. Particularly, I'll tell you about the specific work that Mr. Flaibani, Dr. Flaibani, has been working on, especially during his doctorate studies. This work was conducted at the Evolution Laboratory from the Buenos Aires University, thanks to different sponsorships that he received, or that this project received since 2015, especially for this issue at hand. I'd like to start with a very brief presentation, an introduction. So a flight is understood as an activity that is associated to some specific characteristics of some species, and it has been studied from different perspectives in Drosophila, but in recent times it has been studied in terms of the dynamics using different methodologies. Particularly speaking, I'm going to group them in two big categories. On the one hand, many studies have been conducted with individuals being tied up, as you can see on the screen, let's say. So we have worked with this kind of research that could, as you can imagine, imply a lower, let's say, consumption of energy, because here it is, let's say, this is completely artificial. This method of study is completely artificial. And on the other hand, we have conducted, or some other studies have been conducted under free flight, which are more sophisticated, and we have used, or in order to do so, many different softwares and cameras have been used. On the other hand, I decided to establish a new category, which is, we could say, arbitrary, for some physiological studies where different methodologies were used as well. And here different variables were measured, for example, concentration of enzymes, hormone production, and some others. But what calls my attention, what drives my attention, is what I'm going to talk about, which has to do with the more ecological studies about flight. So when we started to work with this, say after I received my doctorate degree, there was not much information about this. So we decided, I'm sorry, I went ahead too fast. So we decided to focus on the most recent studies that had been conducted. So here we had Fraser and Fraser, Dillon and Fraser and others. So here they were working with individuals in free flight using a dichotomical variant. So we were checking whether they would fly under some specific conditions. So in short, in Diptera, this flight relationship is related to some specific reproductive aspects. And in high altitude, and thanks to the presence of oxygen, the low presence of oxygen, we are going to have a specific behavior, and this is going to impact all of the different biological functions, and it is also going to impact the wing movement. Besides the air density that is going to diminish at high altitudes, it is going to hinder the strength, the aerodynamic strength that they may have. And Dillon and Fraser, in this work that they conducted in 2006, they studied, and they were the first ones to study such effect, these joint effects of two specific variables, where we had, for example, air density and altitude in the performance of this special flight. And they managed to prove that the combination of these two variables diminishes the performance, the flight performance, even more than expected, having in mind an additive effect of such factors. However, I think it has always been considered that insects, that flying insects, could compensate such conditions, that such challenging conditions in high altitudes, they could compensate it with something else. For example, there are many different studies where different characters, different composed or mixed characters have been considered. For example, wing loading, which is a variable that has in mind the body weight together with the wing area, and also the wing aspect ratio, which is pretty much the dimensions of the wings, and also the wing thorax ratio, which has to do with the thorax size together with the wing size. So all of these different variables are considering different aspects, but in general terms, it has been considered that a low wing loading or a lower wing aspect ratio or low and increasing wing thorax ratio would increase the capacity, the flight capacity at low temperatures. So this was evidenced in different altitudes and different places. And it was also possible to see similar results in some specific laboratory experiments under controlled environment. And it was possible to see that they are responding to evolutionary traits together with some others. That's why there are some specific characteristics that have to be taken into consideration whenever we are evaluating this flight capacity. So even though here we're talking about this morphology, very few studies have considered the morphological aspect of it, because most of the work that have been conducted, they have considered very simple variables, such as the dichotomical, such as the ones that I have just mentioned. For example, whether the individual was able or managed to fly or not. And some other times they evaluate the flight time, but having in mind some specific fixed variables. But in general terms, only one species has been used, and they have just used just a couple or very few morphological variables, and they have not even considered sex, or maybe they have, but they haven't, let's say, delved into this discussion. So that's why our study aimed to be more ambitious, but you'll get to see how ambitious it was, and maybe you'll understand better what our ambition was. But unfortunately, given the time restrictions that we have today, and also because of different issues that we have had during the last years, we had to limit our studies. But our main objectives for this study was to study the flight capacity and its relationship with different morphological characters at different Drosophila species, but using a quantitative variable. And also using individuals in free flight, which represents a huge challenge just by itself. And on the other hand, we wanted to explore the genetical baseline of these complex characters in such a way that we could start to identify some candidates as to be later studied, because this is something that hasn't been done before. Now, I'm going to just mention very briefly the methodology we used. And if you have any questions about it, you can just let me know. But in general terms, we work with two pairs of sibling species of Drosophila. On the one hand, the melanogaster, and on the other hand, the simulans. And each one of these species has a specific hosting cactus in nature. And later on, you will see why I am clarifying this aspect. All of these species have a cosmopolitan distribution in the south of Bolivia. The body size is greater than the one they have from their sibling species. And it is also possible to see, or this is something that has been also seen in the body size. And of course, here we have what was expected. It is exactly the opposite for the wing loading. So they have lower wing loading for the bigger species in comparison to their, let's say, to their sibling species. Likewise, we have seen some dispersive capacity, having in mind, or in regarding the other species, the other sibling species. So now I'm going to speak about two specific objectives, two parts of this study. The first part that has to do with an interspecific study, and the second part that has to do with the study of the genetic baseline for the flat capacity. I'm sorry. So in the first part, we work with a stock by each of the species that I have already mentioned, and all of them were coming from, and all of these stocks were coming from a single locality in Argentina, for a single municipality in Argentina. Again, I'm not going to talk about how we collected these individuals or the samples, but in total, we collected 33 different species or 33 different individuals that are coming from the population from another line from the United States. Maybe some of you already know about this species because there's been a lot of publications about this, and many people have published in this panel about this species. So for every stock and for every line, we had many replications, all of them with the same density of individuals, under the same conditions, controlled, let's say artificially controlled in our laboratory. And for all of them, we measured their flight capacity and we conducted a morphological characterization for each of them. On the one hand, after conducting a lot of pilot tests, we ended up having this variable in terms of time for those of that lasted more than 2 minutes, using this device, that is the one that you can see here on screen, as to see how we could measure that, and these measurements were conducted on an individual basis, always in the same range every day, using virgin flies. And they were studied we studied in total 20 to 30 individuals by sex and line. Now, in terms of this morphological characterization, these individuals were, we took some pictures of them, we used some zooms, and it was connected to a laptop, and there we assessed different variables, as you can see in these pictures, using different morphometric measures that enabled us to calculate different estimators, composed estimators, as the ones that I have mentioned, and you can see here, and this and some others. And on the other hand, there were some derived objects as to study the shape of the wing. And so we could see how concentric they were, for example. Something else that I could actually talk about for a long time, but I'm not going to, has to do with the statistical analysis that we conducted for the different variables that we have. And what is important here to mention is that the relationship between each character, each morphological character, and the flight capacity was conducted for each species and sex individually. And also with all of the data collected, we managed to tell them apart from the sibling species. So here we managed to conduct some correction on the data that we used to have. So now I'm going to talk exclusively about the results, the corrected results. So these are the results that that went through this correction, thanks to the new technology that we had with us. So we managed to observe that male flies flew more than their counterpart female flies. So there was a slight difference here on the left, and it was somehow similar in all the cases. However, for the Cactophila species, the greater ones could float more. But on the other hand, it happened the opposite. So the smallest one managed to fly more. So there is always a greatest capacity of flight in the case of female flies. Now, talking about the different studies in terms of morphology and the flight capacity, we only found, or let's say we only had in mind those ones that had been corrected, and specifically for those of the sibling species. So here we have the Cactophila in the middle, and also the corresponding from the other couple or the other pair of species on the right. So you can see how in the Cactophila there was a negative capacity in terms of wing loading and the flight capacity, and with this estimated of the wing torus and the flight capacity, which is what we were expecting according to the results, according to previous results. However, in the second case that we had, it was possible to see a negative relationship for most of the morphological characters. So here you can see just some examples. And in the right-left graph, you can see the negative relationship between the wing loading and the flight capacity, which is a relationship in which we could see greater differences. So here we can see the only case in which this morphological variation explained a greater percentage of variation, and which went around 13%. So therefore, we could see, let's say, a low relationship between this morphological characterization and the flight capacity. So the main conclusions in terms of this first part is that there are some specific differences between sex and species that wouldn't seem to be an estimator, or let's say that this is a very few, or it represents one of the few morphological characters that we usually or traditionally study. And especially in our case, you can see here a scheme of the distribution and abundance of the cacti preferred by these species, which are the ones that we worked with. So here you can see that these cacti, the columnary cacti, are less abundant and they are more dispersed in comparison to the Ocurundium species. So this could explain somehow the greater flight capacity in comparison or compared to their sibling species. And in terms of the second objective, or the research for the second objective, it is worth highlighting that the lines that we work with and the genetic line for each one of them is connected, and actually there is a platform that helps us out to conduct an analysis having in mind phenotype and genotype. So this is useful to identify the different genetic variants that you would like to study in the future. Of course, we work with the proportion of the flying time, but it is also important to say that we have to continue studying this because it has been used to assess the robustness of a variable. Actually, there is a study that hasn't been released or officially released that actually work on different lines, and it is working on a specific variable in terms of a flight capacity and flying time as to check the landing capacity of flies from different devices where they are being launched from. And in this work, or in this research that hasn't been published, here they're working on the association, using the coefficient of landing. All right, so we managed to evidence a meaningful difference, having in mind the flying time, which is what you can see here in this screen. In this slide, where you can see, for example, the relationship between gender and species. So you can see how they cross each other. So this suggests that there is a genetic capacity having in mind a species and also gender. On the other hand, we saw a negative relationship between the proportional flying time and their variation capacity. And this is also going to change in terms of sex as well. So this genetic base would be partially shared, and it would also depend on the sex. Now, in terms of the proportional flying time, we found 53 polymorphisms in 24 genes and 8 intergenetic regions. But on the other hand, for the next one, we could find 80 polymorphisms in 46 genes and 16 different intergenetic regions. So here we could classify them according to that place where they are located at the genome. Either it is a centronic region or an emergenic region, or if it is about a synonymic replacement. So this is a way of, let's say, of interpreting or translating this. So for most of the genes that we managed to identify, we found that there was a unique polymorphism that was associated to the variation in each character. So for the flying time, there was 4 genes that are the ones that we mentioned here in this slide, which showed more than one polymorphism. And there are some others that exhibit more than that. So these are strong candidates as to continue with their study. And in terms of their robustness and the time of flying, they exhibited more than one. So most of these candidate genes are linked to functions that are that have to do with neurological processes, morphogenesis and muscle development, and addition and cellular development. You have 5 minutes left. So for this second part, we could conclude that there is a genetic variability for both species and for both estimators, and the polymorphisms that we detected depend both on character and sex. And even though we managed to see some relationship between these variables, we only detected one gene that is called FAS. In general terms, we can say that we found some interspecific variability for the flight capacity, as well as interpopulational changes for the different species. We also saw some sexual behaviors, sexual patterns or traits for this amongst the different populations and amongst the different species that we managed to evaluate. And in the end, we managed to identify many different candidates that has not been previously identified for this function. So this is all reflected with all of these characteristics, which might be quite complex. So this sexual dimorphism that has not been studied before could contribute to the capacity of evolution of these species and also their adaptive nature, because this is associated to the dispersion that they may have. And given the different proposals and given the different literature that is connected to this morphology and the flight capacity, our results are telling us that the morphology is not necessarily important whenever we are going to explain such variables. So the development of this device and its associated software implied a lot of time and effort in a specific and very difficult context. Particularly speaking, we're talking about something that impacted us all, such as lockdowns and the pandemic, because my 2 biggest projects that I had at hand at the moment actually took me longer and I took a lot of energy from me. But although those situations, we managed to get to some specific and very important results. However, we have been trying to publish these results for more than a year, and we have not managed to do so yet. So, and I think there is some specific bias whenever whenever our peers are assessing this work, actually some people have suggested to publish this research on open journals without giving us the opportunity of getting some sort of discount as to release or publish this investigation. Given that the total budget that we have and that we had and we have at the moment doesn't get to $1,000. And you know that you all know that all of the different publications are way beyond that that budget. So as you can see, I mentioned some studies that I reviewed before conducting our studies. So pretty much we can see how long the studies are taking, how long these studies are taking. The design of this protocol allowed us to go through different journeys, or let's say pathways, as for example, to study the flight capacity of different populations, let's say in different altitudes, which is one of the reasons why I was so moved or so motivated to start this research or to start this journey. We also initiated a panel of study, which is very similar to the one by Belano Buster, which is working with some specific flies that are collected in the same community or the same locality, following the same protocol, following the line of Mrs. McKay from the United States. And we think that this is going to enable us to conduct a comparative study to determine the flight capacity of such a sibling species. And now, thanks that let's say the current situation and that the pandemic is almost over, we could say that we would manage to refine the protocol as to conduct this same research under different conditions and to see their flight performance, this flight performance. With that, I will conclude and I will just show you a couple of pictures from our research group. And you can see here all of our different collaborators, let's say the senior ones and the junior ones that somehow have helped me out to be here. So thank you very much for your attention. Thank you very much, Valeria. This was an incredible talk, very interesting, and thank you very much for sharing your perspectives in terms of everything that you've gone through while conducting your research. So that's why I said at the beginning that this was not going to be a quite conventional or traditional talk, because at the beginning I understood that that was the objective, and as to, let's say, as to motivate a discussion, this group discussion, in terms of the different issues that we might be facing as to conduct this kind of studies in our Latin American region. Yes, of course, that's perfect for what's next, but we still do have some time left to do some Q&A. So we're going to read some of these questions, and we're also going to open the floor for some people if they want to ask any questions. So I didn't explain that much what I didn't have to do in the academic terms, don't worry, but yes, I'm sorry if I was not, let's say, very detailed in some aspects. So here I'm going to start with one question by Carlos Prada. So is it possible to think about a gradient altitudinal or longitudinal aspect, having in mind an adaptation process for this species? Yes, actually, that's what we usually, let's say, think, that the greater the longitude and altitude, the individuals will have, let's say, Drosophila, particularly speaking, but I think that this could be expanded or taken to some other species from different genes, but they are going to have greater body size. But it has to do with the time of development, because at lower temperature, these insects take longer to get to their adulthood life, so they can get there they can get to that point with a greater size. So that's why temperature has always played a key role. And actually, the motivation that I had at the beginning and throughout my doctorate in research project, I saw some differences in terms of gradients, altitudinal and also longitudinal traits. And here there are some other aspects that they do not have in mind. As for example, the ones that I mentioned in the beginning that have to do with the air pressure and then the air density as well. So these factors could actually explain some of the differences that have been seen in morphological lines between latitudinal and longitudinal gradients. I don't know if I'm answering your question. I know I went a bit beyond your question, but yes, you're right. I don't remember the participant's name. I'm sorry. I'm not reading the chat, but he's saying he's thanking you. Yes, yes, of course. So this is what we have, let's say, willing to find, and would love to see how this is all connected to the flight capacity and how this could impact flight in general terms and how this could impact some specific populations of insects. We started to work with Drosophila because it is easier to have at the laboratory, but there are some other species of flying insects that we would love to study too. Okay, perfect. So now here we have another question by Angel Romero. So there were some observations in terms of the dawn time. At dawn, yes. So this is more complex than what I said here, but according to the pilot tests that we conducted, that happened specifically for part of this, I'm sorry, it's not at dawn, but at dusk. So here we have some specific measurements at dusk with some specific species. For example, the Cactophytos species are more active at dusk and also at dawn. So for this Cactophyla species, we conducted some measures, let's say, later during the day, because that's the moment in which flies have the, let's say, the highest activity. I don't know if I'm answering to your question, just let me know. But yes, with all of the pilot tests that we conducted, we wanted to have the best protocol possible for each population and for each species. Thank you very much. Well, there are many congratulations messages for your wonderful talk. And now we have, and maybe we have some extra time for a final question. But now I'd like to open the floor for those people who would like to ask a question, just to raise your hand and just ask your question right away. So far, I don't see any other questions. But in our last slide, in my last slide, I just gave you my contact information, so you can just text me or you can just reach me out as you prefer. On top of that, as far as I know, this is going to be available for any person who would be interested in reaching me out. So there you have all of my information if you want to contact me or you want to contact any of my collaborators. Here I see one final question by Alicia. It says that, have you ever considered having a waiver for the expenses of the publication? We're just running out of time, but maybe we could continue with this conversation in our chat box. Yes, yes. Actually, the answer is yes. And we weren't granted with this waiver because our country is not listed among those countries that require some kind of assistance in that sense. And actually, this is something that I didn't want to mention, but yes, this was something that I was motivated to do, given the discussion that is coming later, because I think that this is one of the common issues that we are all facing in terms of publication. Again, thank you very much, Valeria, and thank you for joining us today. Thank you. I would like to thank all of the participants and also the organizers. Thank you very much. Okay, so to the rest of us, thank you very much for joining us today. Right now, we're going to get started with a 5-minute break. And when we get back, we're going to have a short discussions in breakout rooms, and we're going to talk about the different issues, challenges, advantages of studying evolution in the Latin American region. So let's be back in 5 minutes. Thank you very much.